All right, here are your notes on mitosis in video form. So some of you may have heard of mitosis before. It's one of those things that you may have talked about in 7th grade. Maybe you didn't. Maybe you've heard the word. Maybe you just have heard the word but you've forgotten what it means. Who knows? By the end of class, hopefully you and all your classmates should be on the same page about mitosis. So what is mitosis? By the way, this should be the title of your notes, and these really should go in a notebook. These should not be in a Google Doc because we do need to do some drawing. Also, by the way, you don't need to, but it's going to be ideal if you have some colors with you today. I'm going to be using these colors. So this is just like my normal writing utensil. And then I've got 2 colors that I'm going to use to represent chromosomes, and I've got 1 color that I'm going to use to represent a different cell organelle. We'll talk about that in a little bit. So these are the colors I'm going to use. You can use whatever you've got. If you've only got 1 color, you can make it work. All right, so mitosis. What is mitosis? Well, mitosis is the process of cell division or replication. So when 1 cell becomes 2 cells. And by the way, I'm going to move through this kind of quickly because you can pause the video at any point. So the next thing is, when would a cell want to divide? Well, we've got 4 different times that I want to talk about that a cell would be likely to divide. The first one is when we're healing the body from an injury. So for instance, if you fall and you scrape your knee. When I was a kid, I fell and scraped my knee on the sidewalk so many times. I left so many of my skin cells behind on the sidewalk out in the front of my house. But you need to heal from that, right? You can't just go around for the rest of your life without any skin cells on your knee, right? You need to, your body needs to be able to replace that skin that you just left behind on the sidewalk. So that's one reason why cells would want to divide. The skin cells around that cut say, oh, we have a problem here, let's go ahead and fix that. They divide, produce more skin cells, cover up that area, and you're good to go. If you've ever had a sunburn, a similar thing happens where you damage your outer layer of skin. That's what peels off, and you need to replace that. All these are times that you would want to produce more cells to heal your body. The other one is to grow the body. So if you think about it, you have a lot more cells now than you did when you were first born. You're a lot smaller then. The cells themselves stay about the same size. You just get more of them as you grow up. So you guys are still at a stage in your life where you're growing somewhat. Some of you may be growing more than others, and that's perfectly fine. But since you guys are still growing and I'm an adult, I'm pretty much done growing. You're probably doing more mitosis than I am because your body is still growing. So yeah, when you're growing. The other thing, this is the majority of the mitosis that my body does, is I'm just replacing old cells. Our cells die over time. Some of them live longer than others, and you guys are doing this too. You guys still have cells that have reached their lifespan, and you need to produce more new cells. Yep, we got to replace those old ones when they die. And the last one is kind of sad. It's cancer. I know a lot of us have been affected by cancer in one way or another. I've had quite a few family members have cancer. Some of them died due to it. Some of them are currently recovering. Some of them are recovered. Cancer is something that affects just about everyone, I think. Someone's loved one. And you may have heard that cancer is uncontrolled cell growth, cell division. And that's totally true. In a normal functioning cell, there are processes that say, okay, I've got cells all around me. I don't need to replace any old dead cells. You know, I don't really need to grow anymore, so I'm just gonna hang out and chill and do my thing. Well, when that goes wrong and your cells say, I'm gonna divide, 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 that's cancer. And then you end up with a ton of cells, and they go sometimes so fast that they don't copy properly. So they're even kind of messed up cells too, which can lead to the spreading of the cancer because, the cells are messed up. And when you get a tumor, a cancerous tumor on an important organ in your body, that takes away essential tissue, proper functioning tissue that your body needs, and that's when you might end up dying of cancer. And by the way, I tend to get a lot of questions here about why does cancer make you go bald. And some of you may already know this. It's not the cancer that makes people go bald, it's the chemotherapy. Chemotherapy is a treatment that targets fast-growing, fast-dividing cells. The idea is that these cells are going so fast and dividing that the chemo would target the cancer cells, which are growing and dividing a ton. Well, your hair cells grow and divide a ton as well. To constantly be producing hair, your hair cells are pretty much constantly dividing and growing. So the chemotherapy kind of ends up targeting that as a byproduct. It's just that your hair follicles are also doing a lot of cell division and replication, and chemo just targets anything that's doing a lot of cell division and replication. So that's why people lose their hair when they're going through some cancer treatments. Anyways, mitosis. Next thing I want to write down is the steps that we go through in mitosis. So I'm actually going to start with step 0. We'll talk more about this in a minute. So I'm always going to give you a step number, a name, and then something in parentheses here. And actually interphase kind of has a little bit more of a note here. Interphase is step 0 because it's not actually a part of mitosis, but it's an important part of the cell cycle that I want you to remember, so I like to include it anyway. Now in parentheses, this kind of tells you a little bit about why the step is named this way. So in this case, inter means between. Interphase happens between cycles of mitosis. Okay, let's take a quick look. You do not have to copy this down, but here's a picture of the cell cycle. This is what I'm talking about. So here's mitosis, and here's interphase. So interphase is not really part of mitosis. A cell is either in mitosis or it's in interphase. But like I said, I like to include it because some students then think that cells are only ever somewhere in mitosis, but that's not true. Cells spend a lot of time doing other stuff too, which would be interphase. So just a little bit of a visualization, you do not need to copy this part down. Okay, so the first step of mitosis is prophase. Pro means first. It's the first step of mitosis. Again, I'm going to go through this quickly. Pause it when you need to. Step number 2 is metaphase. Meta means middle. This is not the middle phase. This isn't like the middle step. When we actually draw a picture of a cell in metaphase, you'll see why we call it this. Next up, we have anaphase. Ana meaning sister. Again, this is one, once we actually look at the picture, you'll see why it's called that. Telophase. Telo means distance. Some people, by the way, pronounce this telophase. I say telophase. Another one. You'll see why it's called that when we get there. Oh, announcement. All right, announcement is over. So, like I said, last step, cytokinesis. Cyto relates to another announcement. Hopefully that'll be the last announcement. Cyto refers to the cytoplasm, which you may have learned last year. The cytoplasm is the jelly-like stuff on the inside of a cell. It's what all the cell stuff is suspended in. And kinesis just means movement. So again, some of these don't really make sense just yet. They'll make sense once we're actually looking at a picture of that step. So hopefully you've got that. Let's go ahead and start actually drawing these steps. Now, for each one of these, I'm going to give you a picture. I'm going to draw this picture so you know kind of what order, and I'll explain things. And then I'm going to give you a couple of bullet point notes. So for each of these, try to make sure that the title, the picture, so for instance, we'll do interphase. Okay, so make sure the title, we're going to draw a picture here, and then we're going to have some bullet point notes. Make sure this all kind of goes in the same section of a page. Don't like do the picture on one page and then flip the page and then do your written notes. Make sure they're kind of all together so that you can keep them organized in your notes and then organized in your mind. All right, so let's draw interphase. This will be the most complicated drawing because we actually need to label some things. So we're going to start with a circle. And in that circle, we talked about how our DNA is in the nucleus. So we want to draw a circle within the circle, and that would be our nucleus. All right. I'm going to put this one away, and I'm going to pop my colors out here. So again, I'm using these two as chromosomes, so I'm going to use these first. So the reason I've got two colors is, remember, we have all of our DNA, half of our DNA comes from one parent and half of our DNA comes from the other parent, right? So I'm going to use these to represent both parents. So here's maybe mom DNA is in red, I don't know, and then dad DNA is in blue. And I'm just drawing squiggly lines because, remember, this is in chromatin form. And if you remember, chromatin is our loose, uncondensed form of DNA. Okay? Next, I'm going to take this other color that I said I was going to use to represent a certain cell organelle, and what it's going to represent is these things called centrioles, and I draw them as stars. In real life, they don't look like stars. In real life, they look like little stacks of tubes, but that's more difficult to draw, so I just go with a star. All right, so let's go ahead and label a few things on here. So first off, I'm going to do my little squiggly bracket thing here just to indicate that this whole thing is a cell. All right. Next, I want to draw an arrow right to this inside layer, and that's going to be our nuclear membrane. That's NUCLEAR membrane, nuclear membrane. So it's the inner membrane that kind of makes the nucleus the nucleus. We're going to label the stuff on the inside, our little DNA strands, we're going to label that as chromatin. So that's CHROMATIN. And then we're also going to label these two things centrioles. That's CENTRIOLES. Okay? So there's our labels. There's our image for interphase, and here are the notes I want you to write down for interphase. So the first thing to know about interphase is that the DNA is in the chromatin form, loosely scattered. During this phase, DNA is copied to prepare for the cell to divide. Okay, so we're preparing for the mitosis. And then I also want you to know that the cell spends most of its time in this phase. Again, looking at this cell cycle here, interphase is most of it. Only a little bit is taken up by mitosis. Okay, so get this bit copied down, and we're going to go ahead and move on to our next step. Next step actually being step 1. Prophase. Okay, so we'll start again with a circle. Now the nucleus is going to look a little bit different. In interphase we had a solid nucleus, but actually that nucleus kind of needs to get out of the way. So I'm going to draw it as a dotted line because the nucleus is actually dissolving. The DNA needs to be able to move around throughout the cell. Okay, let's get our DNA in there. Now before we had chromatin, but in prophase we've actually got our chromosomes condensed in, or we've actually got our DNA condensed into chromosomes. So you may remember from our notes that humans have 23 pairs of chromosomes for a total of 46, but it would just be silly if I made you draw 46 chromosomes in here. So we're going to just do 4 chromosomes altogether. I'm going to do 2 red to say we got 2 chromosomes from mom and 2 from dad. All right, so this is kind of like a pair of chromosomes, and this is kind of like a pair of chromosomes. I made 2 big ones, 2 small ones. Doesn't really matter as long as you've got 4 chromosomes, 2 of each color, ideally if you've got colors in your dissolving nucleus. And then our centrioles, last time we drew them both together on this side. This time I'm going to draw 1 on each side. So those centrioles actually move away from each other to prepare for cell division. All right, so there's our finalized drawing. We don't need to label anything here. We labeled everything on the first step. Here are the written parts of your notes for prophase. The DNA condenses into chromosomes, so it had been chromatin before, now it's chromosomes. The nuclear membrane dissolves to get out of the way so that we can move that DNA around, and then the centrioles move to opposite sides of the cell to prepare for division. So pause if you need to. And let's get started on step 2 of mitosis. Which is metaphase. So we'll draw our circle again. Now remember, we said that meta means middle, right? And it's not the middle step. Now the reason we call metaphase the middle phase is because the chromosomes actually line up single file down the middle of the cell. So we still want to take those chromosomes that we had in prophase. We had 2 red and 2 blue, and we want to take those same 4 chromosomes and just kind of line them up. Doesn't really matter where they go, order-wise anyway, just as long as they end up straight in a single file line down the middle of the cell. Okay. Now our centrioles are still over here on far sides of the cell. The centrioles are going to do something really important. What they're going to do is they're going to grow these little arms and attach to each chromosome. And actually that's how the chromosomes line up in the middle, is the centrioles kind of push them into the middle. So I'm just going to draw these little arms, and each one is going to attach right to the waist, we call it, the waist of the chromosome, just the middle of the X. Just like that. So there's your image for metaphase. And you've just got 2 bullet point notes to write for that one. Chromosomes line up in the middle of the cell, and spindles form from centrioles and attach to the chromosome. Okay, so make sure you have that written down. Pause if you need to. Next up, we're going to do step number 3. Which is anaphase. All right, so again, cell is a circle. So this time I'm actually going to start with drawing my centrioles. Okay, so what's happening, let's take a look back at metaphase for a second to understand what's happening. So what's going to happen is these centrioles have all their little arms, their spindles attached to the chromosomes. What they're going to do next is they're actually going to start pulling those chromosomes apart. So some important things to remember about chromosomes is that it's essentially 2 copies of DNA. This half of the X is the same identical sequence to this half of the X. Same here. This is identical to this. This strand of DNA is the same strand of DNA as this. It's just 2 copies of the same thing. Okay. Now, and the whole goal in mitosis is to make 2 identical copies of the same cell. So if I've got one set of DNA here and I've got a copy of that DNA here, I can pull those apart and make those into 2 different cells. Same here. 2 different cells, 2 different cells, 2 different cells, and whatever DNA I have on the right side is going to be identical to the DNA I have on the left side. So that's what the spindles are going to do. They're actually going to pull those chromosome halves apart from one another. So what I'm going to do is draw these little spindle arms only a little ways out into the cell. And then I'm going to draw, and this is sort of the weird thing, I'm going to draw like half an X. So you can kind of imagine if I put these 2 things together, it would form an X, right? But I pulled the chromosome X's apart, so I've got half an X here and half an X here. So I want to do that all the way along, where I had all my chromosomes lined up down the middle. So it should look like that. And by the way, anaphase is called the sister phase. Ana means sister. These are actually what we call sister chromatids. It's where, like I said, they're identical. Okay, so they're sisters, they get pulled apart. Those are sister chromatids. Where the sister chromatids form is the step that we call anaphase. So here we go. This is your 1 bullet point note for anaphase. Get that written down. Pause if you need to. Now, don't get ahead of me and try and draw telophase, because telophase looks funny. I want to talk a little bit about it. Step number 4. Okay, so I'm going to use some silly putty here to illustrate. So far, all of the cells that we've looked at, try to get this into a round shape, it's cold in here. All of the cells we've looked at so far have been round. Well, what happens in telophase is, and actually we can kind of start to see it happening in anaphase, these things are getting pulled sideways. As the cell gets pulled apart, and remembering that the goal is to make 2 new cells, it actually starts to pinch together in the middle and form a shape. It's hard to do. It starts to form a shape, something like this, okay? So those inside bits are getting pulled apart from one another, and the cell starts to pinch in on itself to create 2 new cells. This is actually called cleavage. I know, tee-hee. That's called cleavage. It's cleaving in half. So we kind of want to draw this kind of a shape for telophase. It's a funny shape, but this is how I do it. It's kind of a peanut shape. Kind of with just that kind of pinched in part in the middle. Mine isn't perfect. Yours doesn't have to be perfect, as long as you get that general idea, okay? So, there's that. Now, we've had no nucleus for a while, but in this stage is when the nucleus starts coming back. Now, it's not all the way back yet. We're going to draw it dotted again. Last time the dotted meant it was dissolving, it was going away. This time means it's reforming, it's coming back. So there we go. There's our 2 dotted nuclei in our 2 newly forming cells. So let's talk about what goes in those nuclei. So whatever was being pulled over in anaphase, these bits of chromosomes, that's what needs to show up in each of the nuclei. Nuclei, by the way, is plural of nucleus. It's kind of like octopus, octopi, that kind of thing. So what I'm going to do is draw these kind of half chromosomes in the nucleus over here. Doesn't really matter what order they go in, as long as you've got a couple chunks in there. Kind of like that, okay? And then, can't forget about those centrioles. They're just kind of hanging out over here. They're no longer attached to the chromosome parts. Just kind of outside of the nucleus out there, okay? Short, simple note for this one. 2 new nuclei are formed, okay? And our last step, step number 5, is cytokinesis. So cyto again meaning cytoplasm, kinesis meaning movement. So say we've got, I should have left it, I squished it all back together. Oh, another announcement. Okay, well that kind of let me get my telophase step back. So what's happening in cytokinesis, the cytoplasm, the jelly-like part of the cell, is moving away from one another. So it actually, in this stage, pinches all the way off. Got 2 new cells, and at this point they can kind of go wherever they want. They're 2 independent cells entirely. Okay, so let's draw 2 new cells. Okay. In telophase, the nuclei were coming back. In cytokinesis, those nuclei are all the way back. And they have inside of them, the nuclei have inside of them, exactly what was in the forming nucleus in telophase. So, again, doesn't really matter where they are, just put those kind of chromosome chunks, those sister chromatids, half chromosomes, whatever you want to call them, put those in the nucleus so that they kind of match up like that. And again, don't forget your centrioles. One ended up in each cell. And there you go. There's your drawing for cytokinesis. And here is your one note for cytokinesis. 2 new complete cells are formed. Okay, so there's your finished notes for mitosis. Please make sure those are in your notebook. We're going to keep moving forward with this kind of idea, some of these skills, some of these processes as we move forward. Let me know if you have any questions, and we'll see you next time.